This video is to explain to you how to do the white blood cell count. You are going to need a hemocytometer that has a special cover slip. This is not a normal cover slip. It costs about $25, so please don't lose it. You need a diluting solution, and you need a Europipette tip, and you need a blood sample. What you are going to do is you are going to take the tip, and right here, there's a plug in it. You have to take the tip of this, puncture it through, so you now can go into the reservoir. So again, I took this whole thing, I kept the cap on the tip, I punctured it so it opened up. I now take the cap off carefully, and I have a small capillary tube. So I have a small capillary tube. Do not try to break the reservoir with that. Then I am going to take my blood sample. And to draw blood in, I'm going to squeeze this, open my blood sample, and I'm supposed to fill this capillary tube with blood. So I squeeze it very carefully. And I draw only until the capillary tube is filled with blood. I then suck it in. I then flip this around. I then take the cap, put the cap here, and I then am going to squeeze it so it washes it clean, like so. And the purpose of this is a diluting solution. It is diluting the blood sample by a factor of 20, and it's also destroying the red blood cells. So the only cells I see will be white blood cells. And you are then supposed to let it sit for about 5 minutes while it is lysing and destroying the red blood cells. After the 5 minutes, you're actually going to shake it again and repeat the squeezing in and out. So while I was waiting, I put the hemocytometer on the microscope stage. I have my cover slip on. I am using the scanning power. You might be able to use the low power. Do not try to put it on the high power because it will not fit. And I have found a grid that looks like this. If I go left and right, I have other grids. So see, that's a different grid. I don't want that grid. I want one of these 4 grids in the corner that has 4 by 4 boxes. Now I'm going to load this. So what I'm going to do is I'm going to squeeze this a little bit. I'm going to turn it upside down. There is a little tiny triangle right there. I'm basically going to squeeze the blood into that little tiny triangle. So again, on this, there's a tiny triangle right there. Right there. I'm going to squeeze the blood into it, and it's going to fill this chamber. So I just loaded my sample by taking this, sticking a little triangle underneath the cover slip, and loading it. And this is what it looks like under the microscope camera. I have a couple ghost-like cells right there and right there. That's my white blood cells. I have another ghost-like cell right there. I had another right here until I got it out of focus. This big thing, that's an artifact. It's not a white blood cell. This one right here, it's a white blood cell. That's probably a white blood cell. So white blood cells need to be hollow on the inside. I have pictures of some on the lab handout. So that thing right there, that is not a white blood cell. So what you're going to do is you're going to count. There are 4 of these quadrants right here. So these quadrants are 4 by 4, and they have a known area. So what you're going to do is you're going to count the number of white blood cells that are completely in these squares. And you do it in an S-like fashion. You go this way, down, this way, down, this way, down, this way. Now, unfortunately, when I went to the scanning power, I can't see the white blood cells anymore. So, but on your microscope, it's going to be a different frame of reference, so it should work fine. And you're going to count in each of the 4 corner quadrants. So this is the upper right. I then am going to move it to the upper left. And I actually could see some of the white blood cells here, so there's some cells there. But again, zoom in. So I'll count them again in an S fashion. Da, 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 like so. I'm then going to move the other way. Count it in S-like fashion. It doesn't really matter which S, you could go like that. And again, you're counting only the cells fully in the squares. Then the last one, you're going to count in all 4 quadrants. Now this is so big, I bet you if I zoomed in, that is an artifact too. So you only need hollow looking cells. And if it's in this area or this area and this area, we're not going to count it. This center area down here is used for counting sperm and red blood cells. Because the squares are smaller, it lets you keep track of them more accurately. Thank you very much.